So at the ELCC meeting, we had updated data from the ongoing phase 1 study of AMG 757. This is a BiTE technology, bispecific T-cell engager technology of a drug which targets DLL3. And this is a very interesting compound which is being developed against small cell lung cancer. The BiTE technologies are really interesting because they engage with T cells without requiring MHC, and they do this at CD3. And what that then means is that you get a immune response through activating T cells without being restricted through MHC. And the other end of the compound is linked to DLL3, which is expressed on small cell lung cancer, and therefore can bring the T cells into direct contact with small cell lung cancer to enhance immunogenicity. This is a very interesting area, and we saw data which showed a 14% response rate in a cohort expansion of the drug. It's very early in its infancy, this data, so I think it's a bit early to read too much into the response rate data. The interesting thing was that there was around 40% of patients had cytokine release syndrome, and that was usually low grade, grade 1 or grade 2, but in itself that can be quite tough to manage in routine hospital practice. So I think this is a very encouraging drug that we need additional data on as the cohort gets expanded at the appropriate dose to better understand the safety issues, to better understand the efficacy issues, and therefore the balance between safety and efficacy. And finally, the question is really, do we need a biomarker, and is DLL3 targeting the right way forward? People worry about DLL3 because we used to use Rover-T against DLL3, and this was negative in the Tahoe and Meru trials. But I think it's a reasonable target for small cell lung cancer. The question is, can we use it as a biomarker? And I'm sure that's being evaluated as the trial is being developed further. So it is one to watch in this space, but one where we want to evaluate the efficacy against the toxicities to better determine its role.